So I'm gonna go through radiation therapy, the remaining part from last time. As we go before, like more than half, actually 2/3 of all cancer patients go through radiation therapy. And it actually, we have a strategy, two strategies for radiation therapy. One was, as you remember, there was a previous time about a time effect, which we call as a dose fractionation, because the radiation is proportionally given to the body and accumulate DNA. So what matters is total dose, and total dose at one time is too toxic even for normal cells to recover. So the nice idea from this radiobiology data showing that instead of giving, let's say, 10 gray total, you better to split them given in a fraction wise that would increase the survival of normal cell, while tumor cells, which has a more unstable DNA and DNA repair mechanism, and then we can actually relatively improve the survival of normal cells. So that's where we can have a temporal manipulation of radiation. While the other aspect is actually spatial focusing of radiation. One was the last time we called a brachytherapy, using that specific radioisotope of this one, which will not go too far, and so it's more localized, and you give directly these seeds into, for example, a prostate, so that it can actually treat the prostate cancer. So another way, however, is in general, the radiation, in this case, the high energy X-ray, is so powerful that it will penetrate the tumor, including before and after or normal tissues. So what are the nice idea to how to maximize the dose of tumor, but minimize the dose of normal, which is inevitable. So the key idea is, how about shining the tumor which is residing inside the body? You shine one side, but next time you don't shine it the same way, you shine the other angle. This way, the normal cells are exposed one time, while the tumor got multiple times of exposure. So this is a basic idea. Our idea is very simple. So the question is how to implement it. And that we call it as intensity modulated radiation therapy, or IMRT. So basically, we want to make a device which rotates to provide radiation from angular positions. And also we have to be able to see the inside of the body very clearly. So we do also need a CT scan directly. So there comes this design of an instrument. So you can see electron beam, shine, and you cannot use a mirror like in photon, but this is X-ray. So we have a bending magnet over here. So electrons are magnet bended over here, and beam filters and dose measurement. And we need to go through beam shaping. So X-ray is more like a really directly propagating, okay? So that eventually make an X-ray beam, and that targets the cancer tissue. In this case, it looks like the cancer is inside the brain of a human, or it can be like prostate, internal organ, we can actually locate. And at the same time, on the side, you can see this this another CT scan, not really CT, X-ray, to show the real anatomical feature. Okay, so how do we shine? So while shining this way and the other way, so you rotate this whole irradiating machine. So by this way, and what can happen? So for example, let's assume this is a prostate. Then you shine this way, this way, this way, this way, the other way. While the target is here, let's say if it's prostate, there's a rectum, which you want to save much, because radiation exposure to many times to rectum could cause a big problem too. So you can see this, depending on the dose, you can color code, and you can see the radiation is much more accumulated in that target lesion, while you only have 10 to 20 grades to the nearby normal tissue. So so that was a radiation treatment, and now I want to go to the last time of a systemic cancer therapy, which is a chemotherapy. So let's discuss about what is chemotherapy. We've talked about it a few times before, and the definition of chemotherapy is the use of chemical agent or drugs for their effect in treating any disease, including cancer. So in fact, it's not necessarily only for cancer, but when we use chemotherapy, this is most of the time we like to talking about anti-cancer. So chemotherapy is given a chemical, so most of the time you're giving it into the whole body. So that as a we are talking about that as a systemic therapy. Okay, so what that means is this drug is through our circulation, the patient's circulation. Then, as we understand from transport phenomena, it goes out through the circulation to give out as a diffusion process at the capillary level to reach out every single cell inside our body. So that's why it's called chemotherapy. And of course, there's a limitation that, we want that the drugs go to tumor or cancer only, while this systemic therapy goes to everywhere inside the body. Okay, so in that regard, chemotherapy may destroy cancer cells that have metastasized too. So that's because it's supposed to be reaching every part of the body. But I also showed a movie, which from my own research, that in the end the perfusion throughout the tumor is very important. But it's a real problem that tumors have a heterogeneous microenvironment. Some tumors, for example, pancreatic cancer, has a host character. Is very difficult to diffuse these drugs to get into. So that's like showing as a lowest 5-year survivor. It's almost 95, 97, 8% of death from that cancer. So here comes like what kind of chemotherapies are there. So the idea really comes from cancer is the uncontrolled growth and proliferation. So you watch this general cell cycle. So based on this, many developments are actually targeting any kind of this mechanism. So for example, there's alkylating agents. Alkylating means put an alkyl group, okay? So this will bind to DNA and interfere with replication and transcription. So that binds to DNA. So because when cell proliferate, you have to replicate DNA. And so an example drug is called cyclophosphamide. And this is a famous drug. When you are working in cancer, you hear this quite oftentimes. And this is a prodrug, meaning that this only after it converts into the liver that it becomes a real drug, so that it won't harm beforehand while it was being delivered. Okay, and also nitrogen mustard is also alkylating agent. And another aspect, I think the previous Q&A was talking about cachexia. So many cancer patients who are at the terminal stage, they become like really shrink and do not get enough nutrient, while the tumors absorb most of the nutrient and exhaust them. So what that means, the tumor has always very high rate of metabolism. So why not targeting highly metabolic cells in the body? So that idea comes as an anti-metabolite. So it interferes with a normal metabolic pathway, including when you are making new DNA, it also a lot of metabolic activity happens. So based on that, a number of drugs have been made. So first is the first generation anti-cancer drug, methotrexate, and 5-FU drug, which FU stands for fluorouracil, and gemcitabine. So these are anti-metabolite drugs. Okay. And the other aspect is called anthracyclines. They form free radical and break double strand in DNA, and also inhibit topoisomerase enzyme. So, and this example is a so-called doxorubicin. Okay, so doxorubicin, I think it also has, it's an anti-cancer drug, but many of these drugs are like toxic. I mean, high dose can cause a unwanted side effect, such as heart problem. Okay, and the other is a taxane. So you can see this T. I know that taxane is interfering with the microtubule that starts T microtubule and inhibit function, because when you remember the cell cycle, when the cells have to divide, microtubules are important role to grab this chromosome and grab them and then pull it apart. So, you know, that part can be targeted. So binding microtubule. So that drug is called taxane, and the example is paclitaxel or docetaxel. And these are all like named as a taxol. Okay, so you'll hear a lot about this. Also, there are like a platinum-based drug. So this platinum somehow, a cross-linked DNA. I brought a picture of this. So DNA double helix. Cross-linking of DNA also interfere with the cell replication. So for that, they name it a platin. So it's called cisplatin and carboplatin. And of course, also some antibiotics will generate free oxygen radical that can result in DNA damage. So you can see from the cell cycle, whichever, so there has been many chemical drugs developed which interferes with the cell proliferation. But we have to also consider this, looking at that, is this only for cancer? You know, we are talking about cell cycle. So whatever cells which divide inside our body quickly, or they will be affected. So this is the issue of chemotherapy. So the agent, I'm going to just give you a little bit of detail over here. So alkylating agents. Alkylation is transfer of alkyl group, which is coming from organic chemistry, that from one molecule to another. So alkyl group is CnH2n+1. Okay, so alkylating agents attaching alkyl group to DNA so that it stops tumor growth by cross-linking guanine nucleases in DNA double helix, attacking DNA, preventing replication. And actually, this was being used as a chemical weapon during World War I. So it's known as mustard gas. So, you know, this is a reuse of that chemical weapon into our anti-cancer drug. Okay, 5-FU or gemcitabine is inhibit the use of metabolite, so that we know that tumor cells, once they are growing like rapidly, they suck most of this metabolic activity and energy nutrients in the body. So, so normal metabolism, but you are inhibiting those. So that's toxic to halt cell growth and division. And taxane, so-called paclitaxel, another name is called Taxol, are widely used as a chemotherapy drug. And what they do is disruption of microtubule function, because we know microtubules are essential for cell division. Platinum and cisplatin is a platinum-based drug, and by binding DNA, inhibit its replication. So based on that understanding of chemotherapy, now we can also think of the side effects of these treatments. So what can be the side effect is the question. So in fact, this is like I showed. So you can think of in our body, what parts are keep growing? So you can think of your hairs are keep growing, okay? And as we say, we learned from GI tract. So our GI epithelium is always making a new epithelium. You know that the turnover rate was even like several days. Okay, so that will affect our GI tract and digestion. So of course, these chemotherapy drugs are affecting a lot of our body functions, so appetite loss and fatigue and nausea or vomiting. So you can actually expect this one. So what are the side effects of chemotherapy? So let me tell you a story of my friend's granddaddy who died from cancer. But what he said is this: hey, undergoing this chemotherapy is really like you are trying to kill your normal cell versus cancer cell. So it's a very, very tough and painful. So at the terminal stage, people sometimes say, hey, I would rather die rather to get this chemotherapy to endure all the time. So this is such an unfortunate level, but more than half of cancer patients undergo this chemotherapy. So for example, anemia, the lack of RBCs. RBC turnover times 120 days in human. So we have to keep produce RBC, but this drug will interfere. And depression, even emotional aspects. It hits and fatigue, hair loss, and because our immune cells are also proliferating and keep protecting us while we cannot proliferate immune cells. So that means you have a lack of infection, lack of infectious protection. And of course, pain and nausea, vomiting, and mouth sore. It's part of GI tract. Okay, so very high turnover rate. So what this, in fact, chemotherapy is, all of this is, let me ask you, Kim Yumin, can you guess what this I was trying to say? It's okay if you cannot think of. Okay, where's the Jibom? Ijibom?
Yes, Professor.
Yeah. The same, you can help. Jibom, can you guess? Okay, I'm not asking usually difficult question, okay? So what I mean is, the point is it's non-specific. Are you only targeting cancer cells? No. We are targeting proliferating cells all inside our body. So it's non-specific. That's why we are suffering from this much of side effects from anti-cancer chemotherapy, okay? So that actually, demand us to develop a better ways of targeting treatment. So for example, like this is a total list of body parts which is affected. Alopecia is loss of hair, especially many people, male, female, who doesn't like to lose their hair, right? And mucous membrane, which is a mouth and inside our GI tract, and immunity is compromised, fatigue, weakness, nausea, vomiting, appetite, and also diarrhea. Our gut epithelium is now cannot proliferate. So this leads to digestion problem and even urination and urinary tract. There is also epithelium and anemia, hemophilia. It's a problem of blood clotting. And also, another part, our nails are keep growing and that will be affected. Even nail can be detached, okay? So here comes a picture I want to show how we can actually reduce some of these side effects. So she's smiling, and what she wear is in fact a cold patch to her head. lowering down the temperature lowers metabolic activity and lowers a proliferative potential. So keep it cool down, and she has a finger and toes with a cold water, a cold patch, so that she can preserve her nails. So this is a kind of a way to, during chemotherapy, at least try to save her hairs and nails. Okay, so hopefully this one makes sense. And even blood clotting can be impaired, that we call hemophilia. Okay. So here are these statistics of multiple side effects of chemotherapy. So like this one, you can see from statistics that like 2/3 of patients, complaints about fatigue, hair loss, and constipation. So because of digestion issue, problem, nausea, paresthesia, which is uncomfortable feeling like a pain. Okay. And bad taste, appetite, all sorts of this a lot. So, and even insomnia, the patient cannot sleep. Neutropenia, penia means a lack of it. So neutrophil is become fewer in the blood. So, when you are undergoing chemotherapy, like even before this corona pandemic, you have to wear a mask to protect any potential opportunistic infection. So that's very important because of this neutropenia and thrombocytopenia. So thrombo, that thrombus is for blood clotting, cyto, blood clotting cell, penia, that is a lack of deficiency of a platelet in the blood. So you have to be careful not to injure yourself to get to bleeding, because your bleeding may not stop very well. And hemophilia is inability to blood clot and many other things. So that's kind of something we have to undergo why anti-cancer treatment is so painful and difficult. So there comes an idea. So people, cancer researchers and biomedical engineers, how to come up with a alternative and potentially even better therapy of cancer. So one of them are coming from hormonal therapy. So you guys all heard about hormone, which a lot of them are secreted from a pituitary gland in the brain or medulla. So this is a regulatory substance that is produced and usually transported in tissues, fluids, so that it stimulates cells or tissues of other part, target part into action. So there's a so in some type of cancer are called, the growth is dependent on some type of hormone. So for example, I think a good example is Jang-kyu just did this Q&A session. Okay, this Q&A session, what he said, I actually even put into the text chatting. Triple negative cancer, so which is ER, estrogen receptor, PR, progesterone receptor, and HER2, human epidermal growth factor receptor 2, negative. This is called triple negative cancer, very difficult to treat. And why? Because target is lacking. You can target those specific, for example, ER, estrogen receptor dependent tumor. What it means is the tumor, the cancer cell growth is much on depending on the estrogen receptor, and the estrogen stimulate the cell growth. So our new idea is, hey, what if this cancer cell is ER dependent or estrogen receptor dependent tumor? If you can find that, then you can actually block the stimulation by estrogen by giving estrogen antagonist. Antagonist means that instead of estrogen to bind to receptor to cell signaling to make the cell to grow, you give another one, analog, which bind instead of estrogen to estrogen receptor antagonists bind and block the signaling so that the cells will not being stimulated, cannot grow very well. On the other hand, there's a term called agonist, which is like block, not block, it keeps stimulating, okay? So that's an idea of how we come up with this tamoxifen drug is being used. So that tamoxifen is selective estrogen receptor antagonist, or we call modulator. And this is used to with chemotherapy to treat breast cancer. So some breast cancer is dependent on ER. So, okay. So another, of course, a good example is HER2, which I will talk about later. So because this is a hormone therapy. So for, let's say, breast cancer patient or female, like they may need some kind of this hormonal therapy for whatever reason, but you have to know like the potential of these hormones effect on potential tumor growth. That can be problematic. And another aspect is biological therapy to stimulate the natural function of our eye system to eliminate malignant cells from the body. So remember these hallmarks of the cancer, and our body's immune system is always in the surveillance to watch if there's a tumorous growth and to find out that potential tumor cells and to kill it. Okay, so biological therapy is you want to stimulate our immune system to boost them, to stimulate them so that they can work better as a natural anti-cancer activity. Okay, so here I want to share with you a story of HER2 antibody drug called Herceptin. Okay, so Herceptin is a monoclonal antibody. We will discuss this next time, but it's monoclonal, like real pure, the same antibody against HER2, which is, I said, it's a growth factor, human epidermal growth factor receptor 2, and positive breast cancer. So in the past, when someone got breast cancer, you have a breast biopsy, tumor biopsy, and you do the staining of HER2, and when it turns out to be positive, the doctor will say, I'm sorry, this growth factor is positive. Your prognosis or 5-year survival rate is becoming lower than other breast cancer. However, this can be changed because HER2 positive cancers are about 20% of whole breast cancer patients. Now, we have developed a drug, monoclonal antibody called the Herceptin, and this will bind to this HER2 receptor and somehow block the growth of this type of breast cancer tumor. So now, which respond to this, actually their survival is getting better than non-HER2 positive breast cancer patients, which is great. But however, if you look at the literature, unfortunately, not all HER2 positive cancer will respond to this Herceptin drug. You know how much the drug costs? It's more than $100,000 per year for this drug. And for normal people, this is a way expensive drug. So remember, antibody is very difficult to produce. Okay. However, I have to say explain about the controversy. How, even if it shows some clinical efficacy, potency, but in reality, many times we, the scientists, do not understand what actually is the mechanism of action. So, for example, in this case, ideally, HER2 positive tumor, you give antibody HER2, they will block this activity of HER2 receptor so that it probably blocks the cell signaling of the cell, and then the cells will not proliferate. That's a good hypothesis. How can you test this one? So, you have to have a number of HER2 positive breast cancer cell line. I'm talking about in vitro study, okay? And you give HER2 antibody to the experiment and the other none. And then you want to look at the downstream cell signaling. So for example, HER2 and downstream cell signaling, and to look at whether the signaling is blocked of the HER2. And then it turns out to be, it's not always the case. So then how can you explain this antibody to kill this HER2 positive tumor, even if you block HER2 receptor and it does not block the cell signaling downstream? So that was a mystery. And so there comes, you know, you have to understand like the limitation of this in vitro study. Hey, there could be more complicated mechanism involved because of our immune system. So it turns out that there's potentially another ways of the mechanism of action. So this, let's say, a tumor, let's say breast cancer with HER2 positive, you give Herceptin and it blocks this HER2 receptor. But it may not block the downstream cell signaling, but you realize the cells attached to this antibody will be recognizable by the host immune cells. That because, hey, look at this cell is covered with antibody. There must be something going on. And immune cells come in and then gobble up and kill these cells. So that's proposed as another potential mechanism of action why Herceptin drug is being effective. That we call, in fact, ADCC, or antibody-dependent cell cytotoxicity, meaning that immune cells will recognize this specific HER2 positive cancer cell that was covered by antibody, and that immune cell comes in and eat up and kill the cell. So that's why it's effective. So you see, like this example, even if the drug has been made and showing its efficacy, it works, many times we do not yet know the actual exact mechanism. And why this is important? Because even this treatment later on, there could be a potential recurrence because the cancer cell somehow, you know, everything wants to survive, devise a new ways of different pathway of survival mechanism. So once we understand the mechanism of action properly, then even when there's a cancer recurrence, we could also have a better strategy and idea how to kill and find these cancer cells. So, you know, this is an active area of research, and we call part of this as actually belongs to targeted therapy. So why we discuss about targeted therapy? Because our previous chemotherapy is, as I call, it's non-specific. It just find out whatever cells proliferating in the body, try to kill. But it also kills our normal physiological cell function. So that's kind of a problematic. But difficulty here is, you know, cancer cells are not foreign cells. Cancer cells are also part of our cells. It's not easy to detect and differentiate. We will talk about biomaterials, which is talking about foreign material, which they are foreign, but cancer cells are part of our cells. Okay. Right, because of that, we have been, you know, like people have been working hard for how to only target. So you have to know the cancer more specifically to be able to differentiate. So targeted therapy is blocking growth of cancer cells by interfering with a specific targeted molecule. So this is why I asked you about the chemotherapy, was it non-specific, okay? So these molecules needed for carcinogenesis and tumor growth, rather than simply interfering with the rapidly dividing cells. That is traditional, of course, chemotherapy. Okay. So targeted cancer therapy, you know, this is still our hypothesis, okay? Maybe more effective than current treatment. And, you know, ideally you target cancer cell rather than normal cell. So it's supposed to be less harmful to normal cells. So that example I said is HER2 antibody. you know, HER2 has a various different name. I think you have to hear at least once. It's called new, okay, and it's called ERBB2, okay, and this is a proto-oncogene, meaning that it's a normal gene. However, once there's a mutation of this gene, so that it turns on all the time, that become an oncogene, okay, and that is an example of this. But even Herceptin, you know, if it's high dose, it has a cardiac toxicity. So I'm not talking that even targeted therapies has no side effect at all. No, every drug, any single drug, chemical drug, whatever it is, high dose is likely to involve a side effect. So that's why when we talk about clinical trial, we have to look for this. Okay, so far we have covered the major treatment of cancer, surgical resection, radiation therapy, and chemotherapy. And we discussed a little bit more of a targeted therapy. And so I want to discuss about the challenges and opportunities still remaining in cancer therapy. And you know, this is a time that we talk a lot about personalized medicine, including cancer treatment. And also more broadly, I want to cover about the drug discovery process. Okay, so what are the challenges? So in the end, you will ask, other people will ask you, hey, daddy or mommy or brother, sister, like who are suffering from cancer, when is cancer to be cured? Okay, you know, by definition, cancer is already is the fulfilling proliferation, invasion, and potential metastasis. So it's already very bad stage, okay? But what we have to know is cancer is not a single disease. So this is a word of Professor Bob Weinberg, who wrote that the hallmarks of cancer, and also a textbook of the book, The Biology of Cancer. And I just quote it here for you. Cancer is really a collection of more than 100 different diseases, okay? Each affecting distinct cell or tissue types in the body. What does he mean by this 100 diseases? Meaning that our treatment strategy will be different and the prognosis will be different. So when you look at the statistics of death, sometimes you'll find like cancer, but many times you'll find lung cancer, colon cancer, like different ways of even with the other diseases. So that's why these cancers are quite a different diseases. Now, of course, there are commonality there. So at least, for example, 8 distinct histopathological, meaning histo is a tissue, okay, with a section and watching microscopically, histopathological categories of breast cancer. So when we call breast cancer, that is very generic term, because in terms of if you are in a clinician or like medical engineering point of view, breast cancer have to be self-divided into its subtype. There's at least 8 subtypes, which means like various distinct way. So you have to use different type of treatment. Okay. So each follows its own reasonably predictable clinical course, meaning the prognosis, and exhibiting its own responsiveness to specific forms of therapy. For example, HER2 positive breast cancer versus triple negative breast cancer. There's no use of using Herceptin to this triple negative breast cancer because they don't have HER2 receptors. Right? That's why it's more difficult to treat triple negative, okay? So, and the future comes, I'm sure there are people in our life science department as well. They study, now the era is genome era, like even transcriptome era, and we study cancer diagnosis will be based on bio blah blah rather than the trained eyes of a pathologist. Because in the past or even now we are depending on the pathologist eye, but everyone knows that the time is now changing very rapidly. So we have to base on the information and more of a training and information. So we call it based on bioinformatics. It's a big trend and change. If you are going to work in cancer, you probably also should consider to study these aspects. So the reality of the cancer therapy, especially monotherapy for curing cancer, it's very hard. So the reality is let's say if we find one treatment, let's say one type of chemotherapy treatment, and you hear many of the story is, hey, my grandfather went to chemotherapy and that shrinks the tumor. So we were very happy, like for after a while, 6 months or a year after, we realized the tumor regrow. And this time the same drug doesn't work at all. Okay, so you have to switch the drug and then may or they may not work. So that's recurrence or resistance to a drug. So why, this is the biggest problem why, patients die in the end, because of, you think of a tumor, they keep proliferate. And when the proliferation involves mutations, and so some mutation can provide a survival benefit to the existing treatment, then may reside and then repopulate. And this time that drug doesn't work at all. So that's the limit of monotherapy. So that leads to a new idea of, hey, if you know like this a recurrent tumor's property better, and you realize that monotherapy didn't work, then why not giving a 1-2 uppercut, okay, multiple, punch to this tumor from the beginning, if you know more about that. So that's the concept of logic of multi-drug therapy. So because certain cancer cell phenotypes may be better to kill them through combined action if we know more exactly about the types of the tumor. So several genetic or epigenetic alterations. So this is a nice concept of multi-drug, but I have to say that every drug treatment involves side effect. So it's very likely that, so for example, when in vitro cancer study, cancer cell signaling, you block one part, very specific drug. And then the cancer cells, surviving cancer cells, develop a new pathways of cell signaling or proliferation, and you find that you give a second punch to block that another second pathway. And this way, you could even think of like triple combination, even more. But in reality, all this cell signaling interfering drug will also interfere with the normal cells, and it accumulates side effects. So at some point you realize, hey, again, you are accumulating also side effects. So there comes an issue and limitation. You can't just adding a new drug. So that's a clinical reality. All right. So as we know more about cancers, what are the other challenges? Is discovery of cancer stem cells, which today Jang Gi-ho actually mentioned. Stem cells are very, very few. Major challenges for our anti-cancer drug development. So if we know more clearly about identifying stem cells and their characteristics, maybe we can develop a better cancer stem cell targeted therapy. Also, how do we study our new drug development and treatment strategy is based on preclinical model. And here I put predictive, meaning that, meaningful, okay, of human cancer. So for example, we use a lot of times rodents as a model for cancer, okay? But we are not actually too much interested in curing mouse cancer, but we are interested in curing human cancer. So how we do it is you bring human tumor or human cancer cells into the mouse. the issue is the mouse has its own immune system. It will reject the human cancer. So human cancers cannot grow in a mouse. So how do we overcome this? Hey, let's use immunocompromised mouse. So for example, a good example is a nude mouse. And you may have seen or watched, they usually have athymic nude mouse, which means they don't have thymus. And thymus, the T, they lack of T cell, immune cell. So they are having compromised immune system. Sometimes they can actually grow human tumors. So we use human tumor into the nude mouse and grow and giving our new treatment or new drugs. And on the other hand, like some people who cancer immunologists are very much against this approach because they believe, immune system is so important and you are working with a immunocompromised mouse and giving human tumor and giving drug and then reduce the tumor, saying that, hey, we cured this human tumor in a mouse so that we may want to use it for human. So you have to understand this controversy and we have to admit the limitation. But why we do this? Because that's what's available. Or hope maybe immune system might not be the most important in this case. So that's some of these problem that why we call predictive preclinical model. Means when we are developing a model is always not true reality. Model is only model. So we have to know the limitation and we try to make a model which is more mimicking the clinical reality or even at least focusing on the most important aspects revealing in the human cancer. Also, another aspect of the idea comes from targeting not cancer but stroma. Okay, so why drugs in vitro study kills cancer cells very well, but not in vivo and in human. So there could be many other reasons why, but also there's a kind of a new concept. Anti-angiogenic therapy is a part of the story of concept. You think about it. You want to kill the cancer, but direct targeting of cancer might be a way, but another way is the cancer growth dependent on the vessel formation to feed the cancer. So that's a concept, that a new concept. Hey, maybe we should also consider targeting host stromal cells, specifically helping tumor cell growth. So that's another way of looking at this problem as well. So that we call non-neoplastic stromal cells within a tumor. So that you remember the new hallmarks of cancer, most drug therapies, we also consider why the drugs do not respond. Okay, let me tell you an example of a famous Cell paper in 1999. So you grow human tumor in the mouse model and give let's say HER2, anti-angiogenic therapy. So angiogenic factor is called VEGF. It's so famous, you have to listen many times in literature. Vascular endothelial growth factor, VEGF. So antibody against this actually gave, and then what happens is this VEGF level in tumor goes down, so which is great news. But what we found is not the cancer cell, but host stromal cells, which is non-cancerous cells inside tumor, they actually start to produce VEGF factor because it was like lower, and then these stromas try to compensate for that. So how can you know about this may happen in the human cancer and why maybe specific VEGF blockage, there's a cancer cell and stromal cell, there's an intricate environment in this microenvironment. So and this is something you want to consider, the interaction between this tumor microenvironment. So maybe just targeting cancer cell and cancer physiology might not be enough. you have to look at this whole microenvironment, what's really happening inside. Okay, so that's something I think it's good to consider about it. And of course, all these cancers, tumors are different from person to person. So, ultimately, we, of course, are going personalized molecular medicine, hopefully at affordable cost. And we have to address detailed characteristics of each patient tumor and genetic constitution inform design of a customized therapy. I think this is an active area of research that even like, let's say, a rich patient, for example, you pick the patient's own tumor, take it out, and grow it in the avatar mice, and then give different treatment with the mice to treat, and find out one treatment that works better, then you choose that treatment to the patient. And this is time of like competition of time and effort and cost, but that's even active research at personalized medicine. And now we can see the genetic constitution of this tumor much more rapidly and fastly, so we can come up with a much more personalized way of treatment. However, when someone say, when are we gonna cure cancer? Your answer might be, hey, it's not gonna happen in the near future, unfortunately, because we do not yet know the whole story yet. But what's realizable is reducing cancer to a chronic disease, but bearable. So like diabetes, or nowadays in developed country, HIV is now, I think, as a kind of chronic disease. You don't necessarily die of it because you can actually bear with it. So that might be a more realizable goal. And the future lies in systems biology. Hopefully, this is biological. We consider all this biological response of various human cells, normal and malignant, could be predicted by mathematical models of cells and internal circuitry. When I say circuit, this internal cell signaling pathway, many informatics study the cancer cell signaling more quantitative and holistic point of view, even called circuitry. We don't know everything yet, but this is more like future risk. And I know a number of pharmaceutical companies actually design drug based on this. So this hopefully actually provides us more precise, precision treatment of cancer. Okay, so that personalized treatment, cancer treatment design is in the end, we want to differentiate cancers and normal cells. Unfortunately, cancer cells are part of our cells, but some differences are genetic. Cancer cells proliferate more. Some are more specific. Like cancer cell has subtypes to the type of cancer, even to different individual has different. Okay, when I say HER2 positive breast cancer, that's a subtype. So we both consider genetic treatment because cancer cell starts from one place, use surgery, hopefully early detection is possible, and they divide faster, so we give out radiation and chemotherapy for fast dividing cells. But also type-specific treatment is what I think requires more attention and more of an active research, such as hormonal response, example as prostate cancer, okay, and breast cancer. Some type of these cancers are dependent on some type of hormone. So we can use hormone or hormone replacement or hormone analog modulator to treat or to suppress the growth of specific cancer. And also depending on tissue type, a famous example is thyroid cancer, okay? So, thyroid cancer is a little bit of tricky that the tumor in the thyroid, in some statistics, a female over age 50, more than half does have tumor. So there's a good potential of overdiagnosis. Many of them, they will not develop a cancer, but even our better technology and imaging technology reveals those. So, but when it becomes a cancer, how do we treat? And tissue specific, the role of thyroid, the iodine we intake actually mostly goes to thyroid. So how about like brachytherapy? You give radioisotope iodine, and then the patient ingests, and that goes to thyroid and giving radiation to thyroid tissue. So this is a kind of specific therapy. And of course, a receptor specific, then we can develop an antibody. Okay, so that's something I want to finish up this cancer. And the last part I want to discuss about a clinical trial. So phases of clinical trial. So we call many times bench to bedside, a very nice catchphrase. And how does that happen? Of course, let's say we develop a nice drug candidate. We want to test it. So you can't use it for human right away. So there's lab studies we call preclinical, or even these days, a non-clinical study. And then you go on to phase 1, 2, 3. You've heard a lot from news, like this pharmaceutical develop a new drug. This is phase 3. People are very excited about that. Or what are the caveats and what does that mean? So I have a table here. It's not very exact, but I try to make it simplified. So phase 1, 2, 3, 4, in terms of number and goal. So first, when we have a new drug compound, what you have to do is actually you are using no patient, you have to use a healthy volunteer. Even this compound, you want to give into healthy normal person first by increasing the dose. At what dose this patient feels sick and cannot tolerate anymore. So that's the first thing you want to figure out. So normal healthy, you don't need many, and you can't find many actually. So that dose response it's a phase 1. So I think this is not very exact, but this is good enough, 10 to the 4th. So less than about 10 people, okay? You give dose, and we call it dose escalation. You give more and more and more to see if from normal people how much they can tolerate. To define maximum tolerable dose, MTD, okay? So maximum tolerable dose. So phase 1 is not about real patient. It's only about the drug toxicity, okay? And then phase 2, when it passes through, and then phase 2, about less than 100 people, okay, order of this. And still, you're not focusing on the effectiveness of the drug, but you're more focusing on the safety of the drug, okay? So drug safety and some activity. And that's phase 2. And afterwards, phase 3, now you're talking 10 to the 3. It means thousands of people, huge amount of money and time and resource. And this is the time you are like, you see from the newspapers, right? Now we are talking about efficacy, which means is the drug really effective for curing and treating the disease? But it's comparative, meaning that there comes with the a comparison group, which is already resisting drugs. Okay, after this phase 3 trial, for example, US FDA, Food and Drug Administration, will approve, and this drug is on the market, okay? And still, many times the drug can have a potential problem, which is now we call post-approval phase 4. And that we want to see long-term, let's see, Euiheon?
Yes.
There? Yes, can you guess what I want to write here? Long term?
Efficiency.
Okay, Park Ji-won,
Yeah.
can you guess? Shin said efficiency. It's close, but not exact. Okay, the answer is right over here. long-term efficacy. So this is specific term for, especially for drug or treatment, when they are effective for that type of treatment for this disease. So that we call efficacy, okay? And of course, sometimes we hear a lot of stories about drug withdrawal because even post-marketing, there become a problem. A famous story is the, I forgot the exact name. Yeah, some drug, you know, it's given and even approved. Many people got it, but we found out they give a birth defect for the babies. So we have to withdraw thalidomide drug. So you know, that happens. So new drug is great, but you know, it may not be safe. So we have to consider that. So this is such an important one. I made this a reference table from Wikipedia. I put this underline, which is important, at least for you to know. And there's one thing here I want to show, phase 0, which I didn't talk about, phase 1, 2, 3, 4. Phase 0 about pharmacokinetics, about the drugs oral bioavailability, meaning that the oral intake of drug can actually reach to real target and inside the body, and half-life of the drug. So that's also coming as a phase 0. And preclinical, many of you guys are involved in not clinical study, but preclinical study, because that we use is in vitro and or animal studies. So in the end, how does the drug discovery happens? You know, the name of drug we mentioned about this chemotherapy drug or Herceptin, all these drugs takes so long time to be coming out as a market, okay? So it's good to know how this actually happened. So research and development is first, and preclinical studies, and clinical trial, and review and approval. So research and development, you see the whole thing. For one drug to come out in the market, it takes usually decades and a huge amount of money. So whoever which can accelerate this by, let's say, using AI, okay, or bioinformatics, or mathematical modeling. Okay, these are all very much needed because of how to make it more efficient. So target identification and compound screening and lead drug identification. And preclinical include an in vitro study, which is the easiest, and in vivo study. And however, I have to tell you that minimum 2 mammalian species. Well, what do I mean by that? So like, hey, we test this drug with a mouse model and show it's a good efficacy. It's shown very efficient, but people will not buy it, especially when you are submitting to a big journal. They do require, even cancer research will not accept if you use only one animal model, one tumor model. They do require at least 2. So minimum 2, even not just rodents model, because they can be different from like bigger animal or including human. They also require other species testing, such as rabbits or even dog. Okay, so that's why it takes more time and the standard, because the ultimate goal is whether it will work in human or not. And afterwards, we can undergo into clinical phase 1 to 3 trials and review and evaluation and approval manufacturing. And of course, it requires a post-release or post-approval because there could exist something we haven't seen during this process. Okay, so this is the end of this summary of biomedical strategies against cancer. And I hope this is not only for cancer, but it can actually include other kind of disease. So let me stop it here and then let's move on.